In the 1950s, a young man was visiting a foreign country where his brother was dying of blood cancer. That experience would spark a deep curiosity in the man about blood itself, what it carries, how it behaves, and what it can tell us about disease, leading to one of the most important diagnostic tests in modern history, the hemoglobin A1C test for diabetes.
In this episode, we explore how the very same chemistry that gives us this wonderful test also underlies some of the most damaging complications of diabetes. And remarkably, this chemistry also gives toasted bread, roasted potatoes, and grilled meat their distinctive and appealing flavors. Welcome to episode 4 of the Discovery Dialogues podcast.
Welcome to the Discovery Dialogues podcast, where we take a deep dive into the history of groundbreaking discoveries in the life sciences. Each episode, we will uncover some incredible, insightful, and often just insane stories behind breakthroughs in medicine. Join us as we journey through the twists and turns of scientific history, one breakthrough at a time. I'm your host, Ani Deshpande, and I'm joined by my colleague,
Pamit Kanansari. When we think about major scientific discoveries and their impact on our lives, we often overlook the incredible journeys that led us here. So much of what we take for granted today has a rich history of trial and error, serendipity, and then the breakthrough moments. As we talk about diabetes, I think we have to talk about sugar. It's the elephant in the room. Our relationship with sugar has changed dramatically over time. If we think back to Paleolithic times, if you wanted something sweet, you had to risk getting stung by bees just to get a bit of honey.
In fact, it wasn't until about 2000 years ago that people in ancient India figured out how to refine sugar. And from there, it spread through the Islamic world and eventually to Europe. But the real explosion came in the 1700s when sugar cultivation boomed in colonial plantations across the Americas. While advances in agriculture and industrial food production helped end famine and made food more widely available, there was an unintended consequence, especially in countries like the United States. Food didn't just become accessible, it became overabundant.
Right.
And nowhere is this more obvious than in our soaring consumption of sugar.
Today, the average American consumes more than 30 times the sugar that people did in the early 1700s.
That's insane.
It's hard to believe. But yes, think about all the sugary drinks we have now, sodas, energy drinks, fruit juices that are basically liquid candy. Those are the obvious ones.
But still, it feels to me there has to be more to this story. What are we missing?
Absolutely. There are people who never eat dessert or drink sodas. But here's the thing. Sugar isn't just in sweet things. It's hiding in plain sight, especially in the form of carbohydrates.
Oh, right, carbs.
Exactly. Many foods we eat get broken down into sugar. The most obvious is carbohydrates that are basically just sugar molecules strung together. And today our diets are packed with them. Cereal, bread, pasta, rice, you name it.
And that's what's easy to miss.
And because those foods have become staples in cultures all over the world, especially in modern diets, our sugar intake has quietly skyrocketed, even if we don't always taste it.
And so, what is all this excess sugar really doing to our bodies? To understand that, we need to talk about a fascinating chemical reaction involving sugar that is happening all around us, the Maillard reaction. Let's hear about this from Krish Ashok,
the amazing science educator.
Why does this toast taste better than this white bread slice? Or this golden brown dosa better than a homemade white dosa? Or this golden roasted potato better than this boiled mashed potato? Why is it that above a certain temperature, cooking seems to produce astonishingly amazing aromas, and gorgeous brown color. The smell of coffee, chocolate, the unforgettable aroma of a bakery, grilled meat, or the brown spots on a chapati. Would it surprise you that it's all the same family of chemical reactions, named after a French chemist who had absolutely nothing to do with food, Louis-Camille Maillard.
What Louis Maillard had described, called the Maillard reaction, was sugar reacting with amino acids in proteins. When food is heated, especially above a certain temperature, it attaches itself to amino acids in the protein, creating hundreds of new compounds that give rise to the complex aromas, the golden brown colors, and the rich, savory flavors.
The Maillard reaction is everywhere. It's what makes delicious brown spots on your chapati, the aromatic brown crust on a bread. In fact, the addictive smell of a bakery, the aroma of coffee, chocolate, grilled meat are all thanks to the Maillard reaction. But it doesn't end there. Turns out that the Maillard reaction is not just something that happens in your frying pan to make food delicious. It also happens slowly inside our bodies as we age.
As it turns out, this very same reaction isn't just responsible for delicious food. It also helps explain some of the long-term damage caused by high blood sugar and the science behind one of the most important diagnostic tests in diabetes.
In fact, this reaction is happening inside our bodies from the moment we are born. It's called glycation, a kind of slow internal browning, and is happening in everyone. What differs is the speed. It can be sped up or slowed down.
Now, as we all know, one of the reasons diabetes remains such a deadly disease is that uncontrolled blood sugar highs in diabetic patients can lead to severe complications, including nerve damage, blindness, kidney failure, cardiovascular disease, and even limb amputations, as my own father found.
In the 1980s, scientists started noticing something striking. In people with diabetes, this process of glycation was happening at a much faster rate. They found unusually high levels of what are called advanced glycation end products, or AGEs.
These AGEs started showing up in tissues all over the body, and the evidence began piling up. Impactful studies pointed to AGEs as key drivers of many diabetic complications, nerve damage, blindness, cardiovascular disease. And today there's growing consensus that these accumulated glycation end products are at the heart of what makes diabetes dangerous long-term. Ani, I've met so many people with diabetes, patients, friends, relatives, and what stays with me is the quiet way they carry their fear, wondering if today's the day that their eyesight starts to fade or their feet go numb or kidneys begin to fail, always wondering what damage that sugar may be doing inside, which is why it's so critical to understand not just blood sugar now, but how much sugar your body's been exposed to over time. Because the more sugar in our system, the faster this reaction runs and the more damage it can do.
Okay, so just a quick clarification. When we talk about AGEs, or advanced glycation end products, things can get a little confusing.
Yes, because you might hear some people say that eating foods high in AGEs, like those formed during grilling or frying, is harmful. That is not what we're talking about here.
Exactly. What we are focusing on is the glycation that's happening inside the body, the sugar in your bloodstream that sticks to proteins and other molecules over time.
And it's that internal glycation driven by excess blood sugar that's linked to many of the complications of diabetes. That's the kind of damage we're discussing in this episode. Even though glycation, when it runs amok, can be harmful, the very fact that sugar sticks to proteins where it doesn't belong actually led to the most transformative diagnostic breakthrough in diabetes.
In our previous episodes, we explored how diabetes was diagnosed in ancient times by tasting a patient's urine, watching if ants were attracted to it, or even leaving it out to dry to see if sugar crystals formed. As centuries passed, things got more scientific. In the 1800s and early 1900s, chemical tests based on solutions that changed color when sugar was present gave a simple and effective option for the time, and it helped doctors diagnose diabetes.
Well, thank goodness doctors don't have to taste urine anymore. I don't think my husband would have become a doctor if that were the case. But there was a catch, even with the more advanced color-based urine tests. Sugar only appears in urine after it's already built up in the blood. These tests were only showing you what had already happened. They couldn't tell you what was going on in your bloodstream right now.
That's when scientists shifted to testing blood directly. By the early 1900s, blood sugar tests gave a much clearer and faster readout. But even those had a problem. They could only show what your sugar level was at that exact moment, and that moment could be misleading.
Right. Your sugar might spike if you'd just eaten a cookie.
Or if you hadn't eaten at all.
Yes, in fact, stress, illness, and even the time of day could affect the reading. So a single result could lead to a misdiagnosis. Really, doctors needed a way to see the average blood sugar over time, something more like a record of what had been happening over the past few months rather than a snapshot. In this episode, we tell the story of how that missing piece was found almost by accident. This is the story of a discovery, a test called the hemoglobin A1C test, and how it quietly transformed diabetes care around the world.
Exactly. Today, if you go to a doctor and you're suspected of having diabetes or even pre-diabetes, a doctor will tell you to do the test for glycated hemoglobin or the hemoglobin A1C test or just A1C as it is known. And as our listeners will know, the test is arguably the gold standard for measuring how you're doing on long-term sugar control.
And Ani, in our past episodes, in our quest to understand the origins of discoveries in diabetes, we've traveled to many places around the world. Where is this story taking us today?
Well, the discovery of this test was made not in Boston or in Toronto or in London, but in a very unlikely place. Let us first take a moment to understand why many of the tests that we do in the doctor's clinic are actually blood tests.
Yeah.
When blood flows through our arteries and veins, it's like a river that winds its way through nearly every tissue, delivering oxygen, nutrients, hormones, and immune cells to wherever they are needed and carrying away waste for disposal.
When doctors take your blood sample, if they find clues about infections, metabolic dysfunction, or even cancer, they can take corrective action.
Right. And blood is composed mostly of red blood cells. Those are smooth, disc-shaped cells that flow easily even through the tiniest vessels. And remarkably, these cells are packed almost entirely with a protein, hemoglobin, that makes up 97% of their weight. This protein is what gives blood its red color, and it's one of the most fascinating proteins. It binds to oxygen and releases it to wherever it's needed in the body. And the interesting thing is that there are many different types of hemoglobin. In fact, in the 20th century, scientists started realizing that there were some unusual types of hemoglobin that were associated with human disease. In 1949, the legendary scientist Linus Pauling, who is the only scientist to win 2 unshared Nobel Prizes, he showed that people with sickle cell anemia, a debilitating genetic disorder marked by chronic fatigue, excruciating pain, and distorted red blood cells, had an unusual form of hemoglobin, one that gave the red blood cells their unique sickle-like shape. And this finding, the discovery of the sickling form of hemoglobin, or HbS, was nothing short of extraordinary.
In the 1950s, Anthony Allison, a South African geneticist.
I love this story.
I know, who had studied in Kenya, noticed something really intriguing. He saw that many individuals in sub-Saharan Africa had this sickling form of hemoglobin, much more than in other parts of the world. He wondered why nature would favor such a potentially harmful type of hemoglobin. This didn't make any sense to anyone.
Allison came up with a tantalizing hypothesis. He proposed that this oddly shaped hemoglobin might actually provide protection against malaria, which had ravaged many parts of sub-Saharan Africa for centuries. It's only when you get this form of hemoglobin from both your parents that you develop sickle cell
Right.
anemia. And so, in effect, nature, it seems, had struck a cruel bargain. A single copy offered protection, but two copies came at a steep cost.
Right.
Remarkably, this is one reason why certain seemingly harmful traits continue to persist in our bodies over generations, because in the right dose or context,
Yeah.
they can provide a hidden survival advantage. Even though his ideas were initially considered preposterous, later studies proved him right. And this led to a mad rush in the 60s to study all different forms of hemoglobin. Let's take a moment to appreciate just how astonishing this is. Our blood doesn't just carry oxygen, it carries memories that are etched into it, not just of our own lives, but of those that came before us.
Across generations, across millennia, even a single protein like hemoglobin can archive those stories in its molecular folds. Spectacularly, Allison had shown that studying hemoglobin can unlock the evolutionary secrets of a war between humans and deadly parasites that's been raging for millions of years. And scientists quickly started finding many new variants of hemoglobin that were linked to human diseases that had puzzled doctors for centuries, such as thalassemia and some types of anemia. Suddenly, scientists around the world turned their attention to this mysterious molecule, kicking off what some would later call a hemoglobin fever.
For one young scientist, the interest in blood wasn't just academic. His brother was suffering from a blood cancer, and that personal crisis pulled him into the world of researching blood. That scientist was the Iranian researcher Samuel Rehbar. He was born in the city of Hamadan to a family of 7 siblings, and his father was a garment merchant. His mother was a French school teacher and kept advocating the importance of education for the children.
When Rehbar was doing his postgraduate studies, his brother Parviz was diagnosed with blood cancer, and even though this may seem unthinkable in today's times, he was being treated in the neighboring country, Israel, where Rehbar was visiting him. Despite the treatments, Parviz could not be saved, and his brother's untimely death really guided Rehbar's long-standing interest in studying blood. And there are many stories we hear of personal tragedies that motivate scientists in pursuing a question.
While visiting Israel, where his brother was receiving treatment, Rehbar attended a lecture by Hermann Lehmann. Rehbar was really inspired by Hermann Lehmann, who was pursuing his medical studies, had to flee to England because of the rising anti-Semitism in his home country of Germany, and he moved to Cambridge for a PhD, where a kind couple helped him to get a grant for Jewish refugees and even allowed him to stay in their house.
In Cambridge, he earned a PhD in biochemistry and was given the task of investigating a rampant anemia in British soldiers stationed in India. Tackling this problem would set Lehmann on a path that would make him one of the most prolific hemoglobin researchers in the world.
Lehmann identified anemias caused by iron deficiencies, parasitic infections, and most important to this story, those caused by abnormal hemoglobins. In fact, Lehmann discovered the largest number of hemoglobin variants, and many of them specific to certain regions of the world.
His work shed light on how studying just this one protein, hemoglobin, could reveal deep insights into human disease and diversity.
The young Samuel Rehbar mustered the courage to talk to Lehmann and sought career advice. And Lehmann convinced Rehbar to work on hemoglobins. In fact, he told Rehbar something that he hadn't perhaps fully appreciated before. As you know, Iran, or the Persian Empire, has always been at the crossroads of human civilization, home to people of Persian, Azeri, Armenian, Arab, Kurdish, Turkmen, and Baloch ancestries. And so Lehmann told him that Iran's extraordinary ethnic diversity made it a natural laboratory for discovering rare hemoglobin variants.
Encouraged by Lehmann and inspired by his brother's illness, Rehbar returned to Iran with a new mission: to study hemoglobin variants across the country's richly diverse population. At the University of Tehran, Rebar began collecting blood samples from different ethnic groups, hoping to catalog rare forms of the protein. Little did he know this search would lead him to a totally unexpected area, a new way to diagnose diabetes. In the 1960s, Rebar and his assistants, despite their very limited resources, set about collecting thousands of discarded blood samples. They would wake up at 6 in the morning and go on a motorcycle and pick up tubes of blood from all the university hospitals in Tehran. Soon, Rebar developed a method to rapidly extract hemoglobin, allowing him to screen nearly 300 blood samples a day, and the lab was running like a factory.
Over his career, Rebar analyzed more than 200,000 blood samples, discovering many new hemoglobin variants and giving them uniquely Persian names such as HB Iran, HB Arya, and HB Avicenna.
So in a way, Rebar's naming system was a way to pay homage to his homeland.
Yes, one day when Rebar was looking at a gel and observing hemoglobin bands, he saw something really interesting. He was looking at the blood of a 67-year-old person and saw a fuzzy kind of hemoglobin that he hadn't seen before. And when he looked at the medical records, he realized that the person was diabetic.
Oh, and this is a moment I want to remind our audience that I tell every PhD student that many important discoveries in science are not when you say eureka, as shown in movies and fiction, but rather when you see something odd, something that breaks the pattern, is unusual, and it makes you say, oh, that's weird, or it's unexpected. And following that may turn out to be your greatest discovery. It may not even be the thing you thought you were going to study.
Absolutely. And then he did what any good scientist would do. He tested the blood of 47 other people with diabetes, including children, and he saw, to his amazement, that they all showed the same distinct variant of hemoglobin, which he then called the diabetic hemoglobin variant.
Right. And this allowed him to publish a landmark paper titled An Abnormal Hemoglobin in the Red Cells of Diabetics. What was really remarkable was this paper was entirely from Tehran University, and in it, Rebar had described what he later found was glycated hemoglobin, or hemoglobin A1C. And glycated means sugar is stuck on it. Here's David Dargouzi, a physician assistant who hosts the highly popular Sugar High channel, explaining how HbA1c is different from other types of hemoglobin.
Think of it kind of like this. Here's a molecule of hemoglobin. Now, if it gets close enough to a molecule of glucose, kind of looks like glucose, right? They tend to bond together. So when that happened, hemoglobin just went to becoming hemoglobin A1c. Now, there's a normal amount of glucose floating around in your bloodstream, and if the level is normal, then you would expect that a specific amount of glucose would be likely to bump into the hemoglobin. But if there's extra sugar floating around, kind of makes sense that even more of these are going to get joined together with molecules of hemoglobin. So the amount of your A1C would be higher.
And therein lies the story. Rebar realized that while 4 to 6% of the hemoglobin in normal individuals was glycated or sugar added form, it was more like 7 to 10% in diabetic patients. And you must have realized this is exactly what your hemoglobin A1C test results mean.
Here's David Dargouzi.
So when we talk about your A1C number, if you're in the United States, you've heard numbers like 6.5 or 7.6 or 9.3. And what that's referring to is the percent of all hemoglobin molecules that have glucose stuck to them or glycated. So for example, if I have 100 hemoglobin molecules, and 8 of them have glucose bonded to them, then my hemoglobin A1C is 8%. Make sense?
We now consider HbA1c below 5.7 as normal, then between 5.7 and 6.4 as pre-diabetic, and above 6.4 indicative of diabetes. It basically means there's too much sugar in your bloodstream, over the last few months and your blood sugar is not under control. And it was really serendipity to find that hemoglobin is a good measure for long-term sugar monitoring. The reason is that red blood cells that make hemoglobin have an average lifespan of about 3 months. So when you're looking at the hemoglobin from red blood cells, it's really the average of the last 3 months. And this is what the test tells us.
So finally, there was a measure of what was happening to sugar inside our body for the last few months, not just at the moment the blood test was taken. And this was really game-changing.
But how did the test spread to the rest of the world from Tehran?
Well, it wasn't always smooth sailing for Rehbar. While he was focused on his groundbreaking discoveries, serious trouble was brewing in his country. In 1979, a revolution swept through Iran, which would change the political landscape for decades to come. The Iranian Revolution was a watershed moment in the country's long history. The Shah was overthrown by a popular uprising, and for many scientists and intellectuals, especially those with strong ties to the West, things became uncertain.
Rehbar had collaborated with Western scientists in Europe and the United States and was also perceived to be close to the Shah, and this led him to lose his job at the University of Tehran.
Here, I would like to recall the words of the legendary Persian poet Rumi, who has memorably said in poetic words, and pardon my Persian pronunciation here, Rumi says, Ganj dar virane ast, which literally means the treasure lies in the ruins. It conveys the idea that adversity, destruction, and hardship can often lead to immense potential and opportunity. When Rehbar lost his job in Iran, an opportunity opened up for him to move his entire family to the United States.
He was recruited at the City of Hope, which would become one of the biggest centers for diabetes research, a wonderful institute that we visited for our story on cloning of human insulin in episode 2 of our podcast. And so in the next 33 years, Rehbar worked with a number of different scientists in the United States and all over the world to characterize hemoglobin A1C in more detail.
That fuzzy band that Rehbar had first seen in a 67-year-old patient's blood would become the gold standard marker for long-term blood sugar control.
While having a new test for glycated hemoglobin on one hand was wonderful, it was also the harbinger of something more insidious, about sugar sticking to places it doesn't belong.
It took a while for Rehbar's ideas to gain traction. The hemoglobin A1C test didn't just change how we diagnose diabetes, it completely transformed it.
And it gave us something even more valuable, a kind of canary in the coal mine. You see, glycation, the process where sugar sticks to proteins, happens in everyone. It's part of normal aging. But in people with elevated A1C levels, that process speeds up dramatically.
So when your A1C falls in the range of 5.7 to 6.4%, it's not just a number, it's a warning. It means sugar is accumulating faster than it should, forming those harmful compounds called AGEs.
That acceleration can quietly start damaging our blood vessels, nerves, and organs long before a diagnosis of diabetes is made.
So the A1C test doesn't just tell you where you are, it tells you where you might be headed and gives you a chance to change course before the real damage has begun. The really amazing thing about Rehbar was that even at the age of 83, right until the time he died, he would still go into the lab at 5:30 a.m. trying to understand the ill effects of glycation and how to prevent and potentially reverse these effects. He said, and I quote, "I know that something good is waiting for me in the lab. I enjoy every minute of it." End quote.
Many scientists contributed to the development of the hemoglobin A1C test, but it all began from an unusual type of protein carefully noticed by a scientist working hard in Iran. And there are many people whose lives have been transformed because of his discovery.
Pam, I find it incredible that an immigrant refugee, the son of a Jewish textile merchant from Iran, persecuted in his own country, came to the United States and went on to discover one of the most powerful diagnostic tools in diabetes. His story is really, really inspiring.
I know. And in our next episode, we'll share another remarkable journey, this time of a Jewish refugee who escaped the Holocaust, also from a family of textile merchants. He came to America to invent the technology behind today's most widely used continuous glucose monitor.
I can't wait to tell that story. Please join us for episode 5 of the Discovery Dialogues podcast, where we are incredibly lucky to be joined by the inventor himself.
Until next time.